1 o'clock Eastern time, time to begin. So thank you again, everybody, for joining me. John Quinn, there is my email address and title on the first slide here. This is FlowJo software version 10.10, released 11/20/23, so hot off the presses. I'm going to give you a slideshow overview to try to cover a bunch of things fairly rapidly, and then we'll break out the software. And I've got a few workspaces set up where I can demo the new tools that are in it so that you can see how to use them. I am going to keep the chat window open, hidden from your view, but visible to me, so that I can see if there are any questions that pop up. Please feel free to chat away. I will do my best to pay attention to what's happening in the chat window while we go through so that we can address any questions that come up for you as they occur. Okay? All right. So first question is, I accidentally pulled up some air bubbles at the end of my acquisition. Can you demonstrate how to remove these events? Let's save that for the end. But yeah, that's something we can do. There's actually a couple of ways that you could do that. One, you could run an automated algorithm. So there are a bunch of webinars on QC where we go through in some detail. A variety of algorithms are going to look for differences in flow rate, differences in signal stability, differences just in things that are in or out of the dynamic range, that we can have those algorithms clean the data entirely. I could also show if you just want to cut off, based on event number or time, where those events are. Also good, but I am going to start with the 10.10 stuff. So here we go. And thank you for demonstrating the chat function works and I'm paying attention to it. It's a good start to a webinar. All right, so new to 10.10. And I have to say, this is a nice release. I mean, sorry, I'm like, patting myself on my own shoulder and all that, but we actually got the time and some dedicated resources. So much thanks to Juan Hernandez, who did much of the engineering work on this, to knock out something like 250 tickets. So a lot of those are multiple tickets that related to putting in some bigger new feature, but there's also just a ton of stuff built in where we had time to hit the backlog. And so everybody who's written to me and said, John, I need this fixed, I need this better, I need this different, I think we hit a lot of your tickets. So first, big picture, we've put in spectral plots. We've put in a spectral population viewer. So anyone who's working on a spectral system now has the ability to visualize spectrally what the data look like. We've added advanced scaling preferences. So for the growing array of machines, and especially considering that BD has now put out the FACS Discover S8 that actually includes image parameters now and image derived parameters and so forth. So the growing number of types of parameters one might look at in FlowJo. We've added an advanced features to the scaling so that you can get your data to look just as you want it to look with a lot finer granularity of settings level. Granularity means something when we're talking at the cellular level too. So make sure we're talking about granularity of cells, granularity of settings. Wrong error there. Granularity of settings so that it scales right, perfectly, looks good without any further effort once you set your prefs. We've added SpectralFX, a proprietary unmixing technology. We have added the ability to copy transforms between samples so that if you, modify a transform with some detail on one sample that's part of one experiment that has its own compensation matrix, and you want to apply this to another set of data with a different comp matrix, drag and drop. So you can do that. HyperFinder is now built in, a tool that allows you to figure out the shortest gating path through to a set of cells, things that you have identified, however, maybe computationally, that you would then want to sort on, built in. We've added the ability to create tables for your MEM scores, for your marker enrichment modeling. We have also added the ability to have plots in the layout editor track through population renames. So I think are really useful if, small convenience, a nice thing to do. And then the Cluster Explorer got a ton of work. So Cluster Explorer is a built-in tool that allows you to, as the name implies, explore clusters. Within this version, you have the ability to now use it to look at non-cluster populations, as the name does not imply. So I think that's a good tool to then just generalize the utility of. So we've done that. You can reorder the clusters. You can reorder the parameters. We've extended the heat map views so you can get a bigger panel on the screen. We've made the colors of the cluster names and the heat map correspond to the clusters. So those colors trace all the way through all the different plots that are involved in the Cluster Explorer. And then last, we've also added the ability so you can work on the clusters by merging them and so forth within the Cluster Explorer. Now you can also export anything that you do from the Cluster Explorer back to the workspace as populations so that you can make the things that you do in the Explorer actionable. So all those are the big ticket items. There are a ton of smaller improvements, these that I'm not going to demonstrate, but I will just mention briefly here. We have updated to Java 17. So if your IT department were worried that Java 17 had the best security and so forth, we now use it too. Solves some resolution issues that people were having on high-res monitors for PCs. It also resolves the calculation speed on Windows issue. There were certain operations that involved eigenvalue matrices that were slower on Windows. Fixed it for this by switching to Java 17. The navigation menus are updated and they display more file types. So if you've ever had that moment where you were bothered that you could only see the folder, not its contents, fixed. We've improved the support for spectral compensation matrices for things like the BD A5SE. So just the idea that if your matrix was rectangular, FlowJo didn't allow things like export, concatenate, and so forth, fixed. The n-by-n plots now have a mouse over feature that tell you what parameters you're looking at. So if you're in a giant n-by-n matrix and you were getting lost as to what was I looking at the intersection of these two things, mouse over will do the trick. We've improved the BD CellView Lens plugin integration, including with the BD Research Cloud. So you can now do things like if you happen to be collecting data off the SFX Discover, just add the compressed file up to the BD Research Cloud and it can do the decompression there. And we can also then have FlowJo just smartly download only the pictures of the things that you're looking at in the given population. So a lot more synergy added to those three tools working in combination. We've allowed you to add negative numbers into a compensation matrix. So you could have previously important negative numbers if you had created a comp matrix that had some negatives, and negatives will tend to happen in small doses. You'll tend to see a negative 0.1 now and then in a comp matrix, just because sometimes there's skew in the way a population is distributed around the mean. And actually fixing the skew with a negative number is what makes the MFIs line up perfectly in traditional compensation. So that happened. Now, if you want, you can actually just type in negative numbers as well. Excuse me. Okay, you can delete multiple gates from a group simultaneously. We've added some new bands and organized the bands a little differently. I will show pictures of that. And you can also now set your y-axis scale for the kinetics plots in the layout editor. If you're doing a kinetic experiment and want to set up direct comparisons, we've made it easier by being able to manually define what the scaling should be. So lots and lots of stuff, lots of stuff. There's a bunch more stuff that is even below the level of this that is fixed and works a little bit better. Won't demo this, but there it is on the screen so you can see what should be working better, faster, stronger, or whatever the rest of the Daft Punk song lyrics are. All right. Ribbon reconfiguration. My apologies for anybody who opens up version 10 and the things that they were looking for are harder to find. I believe that you will find this organization after a day or two of using it a little more just intuitive, and you've got better control. I see a question just popped up that said, when deleting the multiple gates, what do you mean by that? So previously in FlowJo 10.9 and before, you could have a bunch of gates on a sample. If you want to delete more than one, cool, no problem. If you drag that to the group starts asking you one at a time, do you want to delete these things? So that was a little bit of a pain, I always thought. And so now we've enabled it that you can delete a whole set of gates from a group at the group level. Well, of course, that's the group level. Anyway, that's what that referred to. All right, so bands. On the cytometry band, you're now going to find, I think I have arrows that pop up, spectral plots and spectral populations right there. You're going to find in the populations band, significant reorganization. We've added the HyperFinder tool there. We've moved a bunch of the algorithm tools out into a new band. So it used to be that populations included like t-SNE, Cluster Explorer, and so forth. And that just didn't make sense to me. I felt like it was time to have an algorithms band. So we do. You'll find all of these algorithms and the plugins drop down in a new band. And we've also taken all the new quad gate tools that were released in 10.9, which used to be parked in the Booleans band, which was, I guess, kind of, and now just gave them their own band. So you'll find those changes in the band and ribbon structure. Spectral plots. So there is both a spectral plots and a spectral populations tool that are built in. So if you look at the spectral plots, you're intending to look at generally one file, and you can look at a bunch of different subsets and so forth, and you can see what the range is across all the different detectors. There's a ton of different visualization tools. I'll go through that and show you there. So built in, just point a file at it, take a look. Spectral populations, pick a population or more than one population, and then you can do comparison on those populations. Both these tools allow gating to happen. I will demo that when we walk through the algorithm itself. If you want to find the cells that were bright in this particular detector and so forth, sure. If you want to compare a gated population with another gated population, you can do so. Spectral populations also allow you to, if you want, associate a specific negative with a given population, so that if you want to do normalization, you can normalize versus a specific negative. Maybe use background autofluorescence or something like that as a tool to use for your normalization. And so these are for comparisons. For the first one, the spectral plots, maybe perhaps even you want to draw some gates on there, identify a particular population that might be a positive and negative control, take it forward to compensation, so forth. Okay. Advanced scaling. So in your FlowJo workspace, there is a heart in the upper right hand of the workspace. That is the preferences. Preferences are the heart of FlowJo, we like to say, and I've also typed it on the slides. We've made that redundant. From within the preferences, there is a cytometer preferences that you can click on, and then this pops up. Okay. Oh, I see a question that says, so spectral plots is only one sample, spectral population is another one. You got it. Okay. So we've got these preferences. You can customize these as you will. They're done on a per cytometer basis. So every cytometer has a $CYT keyword that tells us where did this file come from? What cytometer collected it? So if you clicked on one of these, you're setting the preferences for any cytometer that has that $CYT keyword. Probably you've maybe looked at this, maybe not. It just, most of the defaults are pretty good. We work with the manufacturers to try to get them right. You can turn transforms on or off. You can use this section down here if you would like to filter out any parameters from display. For a lot of cytometers, don't need to. For some cytometers, I'll mention like some of the Coulter machines will have a lot of prominence on the -H, -W version of the parameters, so you'll get every parameter in triplicate. By the time you get to analyzing data, maybe you'd like to hide some of those parameters, so it's a little easier to find the primary -A version of the parameter within your list. Cool. If you're working on the FACS Discover, there are a ton of parameters. You may want to hide a few to make the analysis a little easier. You've got -HW, and now you've also got -T as well. So, there's some filtering you might want to do. But what I'd like to show you is new is the advanced rules. Click on the advanced rules button. This window pops up. So from this window, you have the ability to make as many rules as you want, and you will do something like name the rule, add the conditions. Which parameters should this rule apply to? You can see they're color coded here. So the blue eccentricity rule is highlighting these particular parameters here. The red delta center of mass is highlighting some of those. The green forward scatter, highlighting some of those, etc. You say which parameters these should affect, and then you set the transform right there by hitting the transform button, adjust it as you will. And from here on out, when you add data to a workspace, you can have really, really specific preferences get set. So question, will advanced scaling automatically make sure that the population is already in scale, or do we need to do it manually to adjust it? So what you can do with the advanced scaling is set a specific default for, you can do it on a single parameter basis if you want. So this is nice too. If you want forward and side scatter to have different default ranges, you can do so. And so if the data that you bring in matches your defaults, then it'll be on scale. There is an auto scale option that's built in here. It's actually on the transform tab. So I don't have it on the slides, but we will show it when we flip to the live demo portion, which will then have FlowJo take the first sample that matches that $CYT, figure out what the best scaling is, and apply it to that workspace. So you can have it automatically scale the data each time differently. And up to you if that's what you want to do. I don't know. We've done it. We've allowed that button to be there for people who want to do that. I have it as not being on by default. So you'd have to turn it on if you want that to happen. I don't know if you want your scales to automatically change from one run to the next, even especially if it's just by like a little bit. It's also possible that you will have it auto scale to the first sample, and that won't look great for the rest, that the rest maybe looked better on the default scale. So it's a possibility. You can have everyone auto scale, or you can set in more specific defaults that will line up, fingers crossed, just right with a given cytometer. That okay, my my voltages are going to be relatively consistent, my range, the ability of this cytometer is pretty consistent over time. So fingers crossed, a set of preferences, especially if we can get them down to the individual parameter level if you want to. It should be pretty good just consistently. So you have both possibilities actually. All right. Moving on. So SpectralFX is a proprietary unmixing approach that's on the S8, specifically the FACS Discover S8. It's not anywhere else yet. It's coming to FACS Core, so it'll be available on other things in the future. It takes advantage of the idea that not every detector is the same. And so you can learn some things by profiling the system and adjust your unmixing based on that. And it's available in FlowJo now too. So if you happen to work on an S8, right click on a file, and you have the ability to just hit apply SpectralFX. There are a series of keywords that are embedded in your FCS file that we'll read, and we will modify the unmixing to take advantage of that additional information that you get out of using this new system. So if you happen to have that one, cool. Okay, HyperFinder's built in. Population's band, HyperFinder tool sitting at the end of it. Click on the top level of whatever sample it is with a bunch of gates down below it, and you can end up creating new gates that find the shortest path to that. So what that means is, for example, I've got some file here. Maybe I clustered it. Maybe I used tsne to view it, something like that, and identify some cells that I would then like to sort. But it's really tough to send back to FACSDiva or FACS Chorus the ability to sort cluster 7. Like, what is cluster 7? How do I get that out of traditional gating, which your cytometer is going to need to be able to sort it, and your HyperFinder. So this is the brief view. I think we've got entire webinars on HyperFinder, but the gist is that you tell it which parameters are in game or fair game to use. Like, please, anything fluorescent, sure, scatters, okay, whatever. This is showing an example where we're using some image drive parameters as well, also fair game, since if you're on the S8, those collect those. And then pick the population you want it to find. Okay, so this far downstream population, cool, or a cluster, like whatever you want to do, you put it in. HyperFinder thinks for a little bit, and then it draws gates on whatever parameters are going to make sense to find that population as quickly as possible, and then it draws new populations here using those gates so that these can then be sent back. You can either export to FACSDiva, or you can, I thought I had a slide on it, I don't, or just open a FlowJo workspace in FACS Chorus and use those gates then as your sort gates. So that even if you define some population in a way that makes no sense to us, for I clustered and FlowSom population 1 is really interesting, you can go sort that now. I see something in Q&A, I think. There's a question in the Q&A, what kind of files do we need to bring in for spectral data? Only unmixed data files? It is up to you. Spectral data files can be exported either as already unmixed, and then you can just use them in FlowJo, or you could bring in the raw data and you can unmix them in FlowJo. Your call. I think most of the personal preference aspects of that are like, where do you want to bother to type in the names of parameters and so forth? So really personal preference, but we can also, we can unmix or just handle already unmixed data as well. Okay. Mem allows you now to export a table. For the folks who went, cool, that's all I need for that slide. For the folks who are like, well, what's MEM? Let's go back one moment and just say that MEM is marker enrichment modeling. So the gist of MEM is that you've got FlowSom cluster 1, 2, 3, 4, 5. And you're like, okay, cool. So what is cluster 1? What is cluster 2? And we could go through a relatively arduous process to figure out these are out, figure out which ones are important, or we could click the MEM button. And what MEM's going to do is ask you to compare this population, test, versus some reference. And then it'll just tell you which parameters are the most interesting parameters, which ones are the most highly expressed or underexpressed compared to the reference. and give you a population name based on that. So it's like the 2-second way to figure out what all your clusters are. So I really like this tool. That last term is just an IQR interquartile range, is essentially just a safety net. If one of your either reference or test files has a much bigger just spread of the data, it's a little less reliable, right? And so if those two things are the same or very close, this effectively just goes away. If one is very different, then we can adjust what your mem score is. So that's what that last group, but it's mostly just the difference between the test and the reference normalized on a 1 to 10 scale. So it's also graded. So if you get two things that are really, really different, you'll get like a +8, +9, +10. If they're a little bit different, +1, +2. I find that more useful than just like strict plus-minus designations for populations. So anyway, you hit mem, you get new population names. Cool. The reference could be, if it's a clustering experiment, all the other clusters. That makes a lot of sense to me. If some algorithm have said these cells are a cluster, they're different from all the rest of the cells, hey, compare this population then to all the rest of the clusters and tell me what's the most different. Why did they separate? That feels like a really useful way to use that information. Alternatively, you could compare it to an unstained, a reference, some kind of control population, untreated, healthy, whatever, just to say what's the difference between this experimental reference versus my data. So your call. You have a lot of flexibility with the tool. Now there's just a button, export mem scores, if you happen to want to get these out in a tabular form, as opposed to just attached to the name of the populations. Okay, I see a question that says, do you need the raw data with these files to open successfully? I assume we're talking about spectral data, and we're talking about unmixing it. So if that's the case, and really the answer almost no matter what you're asking about is that the raw data would either be unmixed or uncompensated, I assume is what we're talking about. And you can compensate or unmix in FlowJo and proceed onward. Was probably it. Just seeing what we were talking about, HyperFinder, SpectralFX. So yeah, probably. Sean, if you want to clarify, all ears. Okay, I'll move forward. Keep going here as we are almost halfway through our time. We've got a bunch of extra stuff in the Cluster Explorer. Okay, so you start the Cluster Explorer. You've got this extra checkbox here where you can ask it to use non-cluster populations as items in your Cluster Explorer to visualize, preview them, heat map them, all that kind of stuff. Fun. If you do merge any clusters, you can then also export them as either a new cluster set or the selected clusters. And the difference between that is you can either say, okay, take all my clusters and make a whole new set of clusters that include all the merging that I've done, or just pop this one out as a new population. I found that for continuing on analysis, getting them all out with a completely nicely reorganized naming system, so the new cluster set is probably the best way to do it. If you really are just like, oh, that one cluster is all I want from here on out, then the selected clusters is a reasonably good choice there as well. So I tend to go new cluster set as it keeps things organized and named nicely. But your call. We give people options. Checking back to the Q&A. Oh, it's the same one, and I forgot to click the answered live button. Done. All right, cool. All right. That is it for slides. Okay. Chat box looks good at the moment. So I think, why don't we open up some FlowJo workspaces and do it. I have a question here that says, I hope you will explain how to make UMAP plots. I try but could not plot my data. Your help would be appreciated. So taking a note, at the end, we will remove some bubbles, or at least data affected by bubbles, and UMAP some stuff up. I do not have that planned, but it's an easy thing to add in. It's like a click of a button. And I think the only trick to the UMAP bit is, make sure you've got the UMAP is done as a plugin, T-Sneak's built in. So download the plugin for UMAP. And then there's a couple of choices to make. And I don't think they're overly critical, but we can talk about that. All right, so let's get out of this. Minimize slides and open. Let's see, which one do we want to do first? Let's do the nicely named spectral stuff. Okay, so got some files, got some populations. They are from a spectral cytometer. I can hit spectral plot. First thing that's going to pop up is what detector should I use? Generally all of them, since it's a spectral file, is going to be the right case, and a plot appears. Okay, one caveat, folks. To make this plot, all of the parameters, at least at this point, have to have the same transform. It's tough to like show the same plot and have it make sense crosswise here without the transforms being the same. There's a T button included that if you modify the transform in any way, hit OK, it applies to all the parameters, and then you can look at them on the plot. You can then proceed afterwards by changing, excuse me, by changing any individual parameter, but at least for viewing purposes, that's the way this one has to be. Okay, this is a Mac, so all of my options are at the top right here. If you're on a PC, all of your options will be attached to the window. Okay, and so let's look at what some of the options are. So options, bands, violins, and density are all options. So at the moment, I have density and bands showing. So if I turn off the bands, okay, so there's just the density. So you can almost think of this, each one of these bands is, turn it sideways, how many cells were expressing this intensity in that detector? So a lot of cells had really high intensity in the B810A detector. A lot of cells had low intensity in the V427 detector. So that's kind of the gist of how to read the density. If we just do bands, then we just have essentially averages with some standard deviations plotted. So you get perhaps an easier to read, cleaner feel for this. Alternately, we could do violins, and we could do them with or without the bands. And so that's kind of fun. You just sort of shows the distribution of the signal in each of the detectors. Again, without, I think the density is definitely the prettiest view, and maybe the other views are a little easier to just quickly interpret. Your call, what you like. I'm going to go density and turn off the violins. Okay, so you've got some configure here if you want to do some tuning on how to display this. You can do either the bands, which are showing the percentile on either side of the average line. Density, you could do some stuff like say, you know what, let's trim out like the lowest 5% or something to that effect, and really resolve this plot down to where the cells actually are, take out the outliers essentially. So I tend to come in and almost immediately take a couple of percent off the top of the density just to get a bit of a cleaner view, get stuff off the edges, things like that. So you've got the ability to configure these things. You can reselect which detectors to use here. Under edit, there's some cut, copy, paste, undo, redo type stuff. Fair enough. Display allows you to turn on the grids, the zero line. There's also this population name tool that will allow you to configure how population names are shown. So if we open up this window, we could do something like grab a particular, population over here, say create new, call it what you want. I don't know, call it test. Okay. And then you're seeing what test is. You can turn it on, you can turn it off. And if you want, you can have it named differently based on what your selections are and so forth, so that you could, and we'll see this also gets added right here. Test gates, population test, and then for any other region that we draw, we'll see those pop up. And so there's also an AND gate that shows you the effect of all of your, create new on that one, I don't know, test 2, effect of all of the gates if you want to combine them in some way or another and compare them and so forth. Or go back to the top level. Okay, so some display choices, some go choices, and help if you want to get to the help documents. For those, they're the last of the help documents I'm writing. I have the first help doc on this one going. There's no, okay, I think, sorry, no, these are done. It is the B cell view lens. I still owe one more doc on, and I still have to like edit one more. So apologies if you hit the cell view lens docs and it says in progress. That's on me. Okay, so that is the spectral plots. Spectral populations. We could go down here. We could pick a population. We could pick a second population and say population viewer, same deal with which detectors do we want to look at. Okay, so we've got an immediate comparison going on here of one versus the other. I've deliberately put an autofluorescence signal in so that that's something that we could look at. You can also drag and drop any population in. You can also drag and drop a population in as a specific negative if you'd like to do so. Okay, so we've associated this PBMC's donor with that particular negative. A lot of the tools you'll see here are going to be the same. File, export graphics, options. There's a lot of this, how do you want to show the bands, the violence, the density. One that's different here for comparison purposes is the ability to normalize. And so this is not normalized at the moment. We're looking at raw signal. And so, okay, we've got pretty strong signal up here at the front in the UV446 detectors, kind of on par with the detectors down here. But that's where we have our autofluorescence contribution. And so if we were to normalize this, we could just say positive values if we want. We'd see that's the greatest signal in the autofluorescence is right here at the beginning. And so our blue signal, the one that's coming off of actually our full stained file here, that's going to get normalized versus that negative. And then you can see that, okay, so a decent amount of that signal is just coming from AF, and that makes this peak stand out a bit more. You've got those options. You could also go include it versus negative if you want or not. You could also do the similarity matrix right here out of this viewer. So we've got our various populations looking at a 0 to 1 scale, similar being two identical things, table form, chart form. You can show it across the various detectors. Okay. Display is also pretty similar. The population names, you may want to choose to show only the population or only the sample name. If you only the sample names, fine here to shorten these up a little bit if you want. If you've got very long sampler names that are all the same, help takes you to your help docs. Okay, so built-in tools, ability to set some things up for compensation purposes in the future, and the ability to view anything in a variety of ways spectrally. We have a question on how can you rename the sample once it's in the workspace. Oh, well, quick answer live, done. So the sample name, the thing that appears under this name column, is actually just a keyword, and it could be any keyword, and it could also be a keyword that you make up on the spot. Your preferences include workspace preferences. So each one of these tabs is a different part of the program, and nearly everything in FlowJo is customizable. So if I hit workspace prefs, you'll see that I'm using the short file name. The default is the FCS default, so the sample name keyword, whatever is appended in your FCS file. So I like the short file name so that whatever the file is called, where it's sitting on my desktop in this case, or if it's on my server, that's what I see in FlowJo. But you could also pick any keyword. So if you were to open up one of these files, inspect it, inspect just tells you everything there is about a file. There's this whole list of keywords on the left. If you were thinking, well, the name that appears here isn't the name that I want, you could scroll down this list and see if some other keyword has the information that is most useful for you to name it, and then select it in the prefs as the thing to use right here. Say, please pick that keyword or combination of keywords. If nothing there is what you want, you can use the workspace tab, and there's make a new keyword. Make a new keyword, type anything you want into the column, and pick that new keyword as the name, and that's what will be the names of your files, and that'll be consistent and carried out to the rest of the program. So the answer, as is many things in FlowJo, is a preference. All right. Going back to show all. Hey, we were heading here next anyway. On my list of things to make sure I showed you. Advanced scaling parameters. I'm going to, so let me click on this file, because that is off the S8. And so that'll have a bunch of fun extra parameters. I'm going to hit cytometer. I'm going to pick, happen to be picked there anyway, but whatevs. It's the $CYT discover. So it's showing how I've set this up to filter out some things, etc. The advanced rules button is what I want to get to today. Clicking advanced rules brings up this window that I showed in the slides. So here we are. We've got the forward scatter rule, and it's got a condition. Parameter name contains FSC. And we can see which parameters are highlighted, have the blue square next to it saying when we went through these parameters in order and figured out which ones they should apply to, these are the ones that match. You've got a picture of the transform. This is how I want it to look. Cool. I've got one set specifically for eccentricity, which has a 0 to 1 range, which is kind of weird compared to some of our other linear scale parameters like forward scatter. You wouldn't want to be on 0 to 1. So that's nice to have the ability to control this parameter very specifically. I've got my fluorescent parameter settings here. So this can be, this is actually an interesting one in that amongst the things you can do is adjust your positive decades as a tunable here, whereas that's not something that you can typically set in a preference because the positive decades works together with the range of the data. And so it tends to adjust automatically. And so if you wanted to set one of these things that have, say, a particularly high range to also use a specific number of positive decades. You couldn't have done that in previous versions of FlowJo. So that'll matter for Facts Discover. It'll matter a little bit for Aurora data, which has a range that's just about there. Matters a lot for like the Quantion, the Novasite, some of those that actually have like the 7, 8, 9 decade at least displays, but are still only going to use 5 decades. So, FlowJo assumes by default if you're showing a top of 10 to the 9, then you have a huge range. If you're actually still just really going 10 to the 9 to 10 to the 4 or whatever, it's nice to be able to tell it how many positive decades and set it here. So there's a bunch of cool stuff you can do with this. Why don't we make a new one? I'm going to say add rule. And the one that I had mentioned is what I'm going to different forward scatter and side scatter scaling. So I could call this rule whatever I want. I'm going to call SSC for side scatter. For conditions, I'm going to say add condition. There's a bunch of things you can go with. Parameter is side scatter would work, for example. I also just show you what if you want to type something in. Parameter name contains, I'll say just contains, and also SSC. So you'll see it's going to highlight the ones that match my condition there. So they're green. You can do multiple conditions and just notice that you can do and or by just double clicking on this. Since I only have one, it doesn't actually matter at the moment. Then I would say transformation and say, oh, I don't really like that transformation. I want side scatter to be bi-exponential. I want it to be linear. And for this particular cytometer, I want it to have a much bigger range than what was showing up there by default. Maybe something like that. We'll do that and say, okay. So we've just made a new advanced rule and that's remembered. You can see that there's 4 now. Say, okay. The next time I add data that matches that $CYT keyword, it'll follow all those advanced rules at the time that it loads into the workspace. So you don't have to do anything further. If you want to tune some things automatically with the T button, you still can. But it's also, if you've done a good job with deciding what looks nice in that advanced scaling window, things will just look right off the bat. So cool. Last thing to show in this works, and sorry, I'm getting over a cough, so my voice sounds like this, and I'm going to go every few minutes. We are better though. All right, so I've got this SpectralFX data here, meaning it was just data that was collected on the S8, so it has the SpectralFX keywords built in. I right-click on it. I say apply BD SpectralFX. That is going to think for a moment. It's going to, if we see the color of that matrix, it's going to get a new compensation matrix that's going to say acquisition defined, BD SpectralFX. Probably needed to change the scaling on this one a little bit because stuff stuck to the axis there. That's okay. And that is going to then, after it's done thinking, and like it might already be done, it's going to then have used the SpectralFX keywords to create an unmixing that again is reflective of the characteristics of the detectors on the instrument like as it was when you collected the data. So it's pretty custom to each time that you run it. That's just a nice way to do it. So no big display here. Some of the effects can be fairly muted. I mean, if the data was good to begin with, there's only so much better you can make. This was a pretty good data file. But I think that's, if you happen to be an S8 user, you'll find this is a really nice tool to have built in. Okay. Check. Moving on to another workspace. I'm going to minimize this one. I'm going to open up the one I call copy transforms for a really simple, quick demo. Simple is good. Okay, so I've got 2 different data files here. I have actually taken this matrix, the acquisition defined, and just duplicated it. So they actually have the exact same matrix here. So I'm kind of faking this a little bit, but just imagine that you had 2 data files collected at different times, and you wanted to take the transform. So you've bothered to adjust the width basis, the range, anything like that on one of your data files. FlowJo is smart enough to know that if you've got a series of files that all share the same compensation matrix, if you adjust the parameter on one, will automatically adjust it for everything that shares the same compensation matrix. If they've got the same matrix, they're probably part of the same experiment. You want to do direct comparison. This just should happen, and it does. If you've got 2 different matrices, however, we're not sure it's the same experiment. In this case, it is. I modified one slightly, or in this case, not at all, but that's okay. FlowJo thinks I've modified it because I duplicated it. And I go into this one and I say, let's take C38 and let's customize the axis a little bit. Let's drop off the extra negatives. Let's make this width basis a little smaller, say apply. Zoom controls are in the way. Move that. So if I go to the other file, it's got a slightly different transform. If I want to make those transforms copy from one to the next, I just drag and drop one file to the next. It'll ask me if that's what I want to do. Do you want to apply the parameter transform settings to everything that has this other matrix? If I say yes, done. That's it. Transform settings copied. So I feel like that is a small tool that will help just save a ton of effort. If you've gone through and maybe tweaked 3, 4, 5 different parameters, perhaps on like a 20-something color experiment, you'll make sure you do all the settings exactly the same. It's a nice way to go. All right, there's that. How are we doing? We're doing okay time-wise, I think. Let's go here. So here, oh, sorry, UMAP person. I already ran the UMAP, but easy enough to fake the demo. I've clicked on the things that I want to apply UMAP to. Could be particular level of the population. Maybe I wanted to do it on the T cells, whatevs. If I apply it to T cells and everything upstream of that, if I were to show it on the UMAP plot, if I would say UMAP the top level, they would get a 0, 0 value for the UMAP. So it's fine. You'll just only see the things that you've UMAP. So choose which level you want to do it at. Probably takes a little longer to do it for more cells. So pick the one you're actually going to work on. And UMAP is not built in, it is a plugin. So I would click the plugins dropdown. I have gone to the FlowJo Exchange. So I'm going to FlowJo Exchange here. It's just going to bring up the FlowJo Exchange just to say if you wanted to get UMAP, internet working at the speed of, I don't know, not light. It is Fios. Anyway, sorry, the plug. If I wanted to get UMAP, I could type UMAP. There it is. I hit download. I have it. I unzip it. I put it in my plugins folder. And then plugins. UMAP is down here in the list. It's alphabetical. Okay. I pick which parameters I want it to factor in. Probably it would just be the compt. And even more so, if I had done some gating upstream, like I've gated out the live dead. I don't really need the dead marker in there. If you add additional parameters that are kind of all the same for all the file, like I only have things that are negative for the live dead marker. Anything that's the same for all the files tells these kind of algorithms that tries to put like things together and different things apart. If you've got some parameter that's the same for everything, is push everything into one pop in the middle. Not helpful. So I'll leave that one out. I would pick what kind of scaling I want to use. Euclidean is one that most people are familiar with. Cosine distance tends to work fairly well with this too. It's just measures of how far apart things are. So either one of those is fine. You can really pick any of them. Those are probably the best. You've got some tunables for nearest neighbors, minimum distance, etc. So these are just tunables that are going to do things like, should I make my populations overly compact, or like should they be really tight populations? Or do I want to spread them out a little bit so that I can see a little bit more of the nuance? That's just the minimum distance. Nearest neighbors is how many nearest neighbors do I want to look at? So you can play with that. A bigger number looks at more of the data, of course, gives you a more global representation. A smaller number looks at only things that are a little closer, gives you better local resolution. So there's some give and take for that. Just choosing the default is usually a fairly nice option. Say okay. It thinks for some amount of time. I think it was like 3 minutes or something for whatever, 400,000 cells or so here. And then it pops out 2 new parameters, UMAP1 and UMAP2. Add some gobbledygook numbers at the end. So if you want to run it a second time, we can add a few more gobbledygook random numbers to have 2 different versions of your UMAP. Cool. And then if you double click on a population, hey, there's my quad gates. UMAP parameters just appear on the list. And then we can use them like any other population, or sorry, any other parameter in FlowJo to visualize the data. You won't know immediately what these populations are because UMAP 1 and 2 have no biological meaning, but the cells in a given island tend to be of the same phenotype. They've grouped together because they express similar markers, and that's where the cluster explorer comes in. We can use a variety of heat mapping tools to do this. We could use MEM to do some naming, et cetera, et cetera, et cetera. There's a lot of ways to go about this. Cluster Explorer is a nice way, and that is the one we've done some updating for. So let's talk about it. Okay, I've got FlowSum clusters run, UMAP run. You don't have to have UMAP run. You do have to have clusters. Well, actually, I guess you don't have to have clusters in there. Duh. Part of this was I was going to tell you is you could just run it on, I don't know, set a quad gate, something like that. Pick the parent population, click the Cluster Explorer button. Cool. If you want to show populations, not necessarily clusters, you could hit the configure, you could pick those quads, accept that. Which parameters are you interested in get selected here? I'm going to leave out the dead marker, not so interesting. Do you want to create a map showing your your populations on some kind of set of derived parameters, such as UMAP or t-SNE, et cetera. Let's use those UMAPs. And I've only run clustering algorithms, so that's what's selected here. But if you ran more than one, you could also compare results between two different clustering algorithms and get some Jaccard similarity coefficients. There are other webinars on clustering and so forth. But moving on from that, I will accept. That's going to think for a sec and pop up a whole bunch of different views of that data. There is not a template for Cluster Explorer. It is actually an interactive tool. So it works a little different every time you do it, depending on what data you have fed into it. Okay, and this one is stuck behind my Zoom tools. Can I move this? Oh, awesome. Well, okay, that makes it harder if you're using Zoom simultaneously because the Zoom controls right in front of half of my controls for the controller. What is? What we've got here is a line plot, bar chart, UMAP view, so any kind of 2D plot is fair game here, and heat map chart of all of our clusters. Things that are new, the colors of the clusters now line up in the heat map with the lines, with the bars, with the clusters here. You can reorganize if you're like, let's see, I'd like to put this one here because it's got a lot of blue in it or whatever. Cool. And then you can see that the plots are going to rearrange to do that. So that's going to make some sense there. We could find the green population and the red population. You can also rearrange the clusters. I'm going to highlight green and red and say, so these two populations, I'm not sure I totally believe that green one is different. I can look over here at the plot and see that there's a bit more HLA-DR in the green one. So maybe they should have been two, but for my purposes, they look like they really should be one. I'm going to go to clusters and I'm going to merge them, merge selected. There we go. We've now just got this cluster 11 instead of 0 to 9 and one more, that we've put those two things together. Maybe it was a bad job by me. I think these, even by eye, that really stands out, that maybe that the HLA-DR was a separating factor, but never mind, pushing forward, assuming we've done a nice job of merging them. So we've got that merged population that's in there. If I want, I can go back to clusters and say export, and I can export them either just as the new selected cluster or the new cluster set. I'll do that. Done. So they've been sent to the workspace. When we look back at the workspace, we'll see we've got a new cluster set that reflects the fact that instead of having 10 clusters, we now have 9 because we've merged these two, and they are one population that shows up in all the heat maps and bar charts and all that kind of stuff. Okay. Other thing I should show is if I can get to this, and I think I can. This says behind my Zoom. Oh, I can drag it down. Good. Yeah, I made it. Okay, that's it, done. Right here, what are we looking at? We're looking at T cells, that parent population. Because I've selected just these gates, I can show the individual gates and so forth also as just how did the cells in each of those gates fall amongst all my clusters. All right, so there's that. Okay, reorder, expand the heat map. Okay, so that's just saying that we've done this here. You don't have to scroll anymore. Non-cluster pops, cool. Okay, I think with like 10 minutes to spare, I think that is the bulk of what I wanted to show you. I think we're okay here. That'll allow me a little bit of time. We can talk about, I think I showed the UMAP bit. Let me talk a tiny bit about what if I wanted to remove bubbles. Okay, but first, questions. Questions on any of the new stuff? Looking at the chat, we are looking like we're doing okay. Looking at QA, looking like we're doing pretty good. Perfect. All right, I'm going to close this up then. Oh, I didn't show the mem bit. All right, one more thing. There's my new cluster set, by the way. CE cluster set, cluster explorer set. I'm going to come down here to my FlowSum populations. Ooh, okay, there's my FlowSum populations. The mem tools right here, marker enrichment modeling. I can create mem scores on these. So again, it's telling me which parameters here are fair game. Almost all these tools start with which parameters do you care about? I'm going to care about pretty much all of them most of the time. Not so much about live dead, that's fine. Here's the bit of select what kind of reference you want to use. So mem auto reference means all of the other things you've selected. So I've selected 0 to 11 here. And for each one population, everything else is going to be the reference. So this is good for clustering. You could also do a user-defined reference. That's where you maybe will pick like an unstained or something like that, or an unstimulated if you're trying to do some kind of comparison between experimental conditions, or referenceless mem where you're like, not totally sure. Just start with the left side of my data range, call that the low numbers, the right side's the high numbers, and interpolate between. So I'm going to do auto-reference for this case and say OK. So the MEM algorithm is going to do its thing. And in a moment, we will end up with MEM scores, append it to the names of all these data files. So we can say things like, what makes population? I didn't show HyperFinder either. Why didn't I write that down on my list? All right. No, I did. It was just on the left side. Two more things to show. Okay. So one here is like, okay, so population 9, really, really brightly expressing HLA-DR9. We could also, not that this is new, this is from in 10.9, but I will show we could also change the MEM threshold. If there's a lot of +1s in here, maybe we're not super concerned with things that are minorly brighter or dimmer than the reference. Let's say you have to be at least a 2. Okay, so that gets us right to things that are really, really much more different. And so maybe like, I don't know, that cluster 7 is pretty interesting. Interferon gamma +7, CD8 +7. No, sorry, BD is going to ask me for a password because why not? Okay, so we've got that stuff. The part of the MEM stuff now that is new is that you can go to the bottom and hit export MEM scores and pick your file format. You want the node names with the MEM scores, the original names, do you want both? I'm just going to put that on my desktop and export. And I'm going to get a CSV file that has my MEM grid. Cool. I will assume that it is okay to believe that a CSV file can be opened and read. We don't need to do any further on that. Okay, we have some questions about the quad merging tools at the end. Yeah. In fact, I'm just going to say that right now, since quad merging, I believe you assume is in MEM. So the idea here is that for our MEM scores, there is a merge nodes based on the MEM score. So the MEM score is trying to tell you what it is that this cluster is. And so if you end up with 2 populations that say have very similar or exactly the same MEM scores, maybe those should be joined into one cluster. So whereas I merge the clusters sort of manually by inspection in the cluster explorer, I could do it mathematically here using MEM scores. You can imagine that since your actual data tends to have a range of like 262, 144,000 bins, number off the top of my head, that if you simplify this to like a 0 to 10 range, which is what MEM does, there is some room in there where you could say 2 things that have a little bit of variability might end up with the same MEM score. And so they may have clustered differently, but should be put together. So if I do that, merge nodes on MEM scores, this number here says, what's the biggest difference I can allow between any one parameter's MEM score and still call these the same thing? I think the only reasonable choice to type in here will let you type 0 to 5. But I think it's either 0. The 2 things better have the exact same MEM score if we're going to merge them, or 1. We're going to let a little bit of variability go in. Anything more than that, you end up with like, hey, all your clusters are the same, I think, really. Let's go 0. Say OK. And so what happened here, see that not merged appear on any cluster that was left alone. Ones that are lacking the not merged means that looks like population 1 and population 5 were probably merged together. And also these 2 that are merged, MEM scores less than marker inclusion threshold. We're going to look down and we're going to see that there are some Boolean gates. Let's scroll a little bit. Okay. Some Boolean gates. Did I not get any? Oh, maybe none of them were merged and we just had a name that was too long. Just expand that again, make sure. There they are. Sorry, they were just off the bottom of my screen. So clusters 2, 3, 6 were merged, and we have the originals up here, anywhere that it says that it doesn't have the not merged, and the Booleans there. So feel free to proceed with one or both, however you want. So I think that's a really nice tool. I'm showing a relatively small experiment here to move through this in an hour. But if you had a bunch of clusters, 25, 30 clusters, whatever, you may not want to walk through one at a time and try to determine which one should be merged. So I think that's a nice way to potentially simplify if you've maybe let the first pass of clustering be pretty broad, make a lot of clusters, see what makes sense, and then use MEM to put them together. Reasonable choice. All right. Last thing here, and then we'll try to hit the questions that came up. We've got 6 minutes, but HyperFinder is relatively quick to show. So I'm going to pick the top level population. I'm going to say now I want to sort on, let's see, cluster population 6. That was pretty interesting. A lot of interferon gamma, a lot of CD8. Actually, 7's even more so. Interferon gamma plus 7, one of those two. Either one is fine. I'm going to click the HyperFinder button. So that's going to pop up. And I wrote some documentation up here that's going to explain in more detail some of your options. We'll go quickly here and just say you've got a few different types of graphs you might want to be able to do. Polytope is a nice one. You can set a limit on how many gates it can use. So in case you're on a Diva version that only allows for 8 gates, cool. Say don't use more than 8. Tell it which parameters are fair game. I'm going to say all of the parameters. Scatters, fine. Ooh, why not all the scatters? And you know, live dead, sure, because we don't want dead cells in there. And which population we want to find, 6 or 7 were going to be fun ones. Let's go 6, let's say okay. And Hyper's going to think for a bit, and it's going to have this building gates. After about, I don't know, 30 seconds or so here, it's going to start showing us pictures as well to illustrate what gates it's building. And then it's going to use what we'll see will be green, as it'll take that population we said, find this one. It'll take most of it, like 80% of it is. And it's going to say, let me draw a gate around those cells. And then it's going to isolate it, a test set. So it's going to use for validation the red dots that are going to pop up on the screen in a second and say, okay, if I drew the gate right around the green dots, do I also get the red ones? Is this generalizable, or am I, overly fitting this to the data? So it breaks it up into cells you want, cells you don't want, and cells you're testing to make sure that you got. And it gives you an F measure score, which is the harmonic mean of precision and recall. It's like, did I get all the ones that I wanted? How many did I miss versus how many that I didn't want that I happened to put in? So you can see that the F measure score is updating as we draw this thing. 1.0 is perfect, so we're going to end up with a really good F measure on this thing. Let me check the Q&A while that stops. Is MEM score a non-user bias way of merging than cluster explorer you merged earlier? Yeah, good question, and you are absolutely correct. Whereas when I picked the green cluster and the red cluster, I was like, well, they look kind of next to each other. I'm going to merge them. I had the ability to look at a line plot, and I could see for each parameter, was there a parameter that those two clusters had some differential expression? And in fact, we saw it. It was the HLADR parameter, looked very different. And I merged them anyway. So user bias was totally in play right there. You can check yourself versus your graphs and all that kind of stuff. If you want to make it less biased, you can have your own rules of thumb. But MEM score is just math. And so it is unbiased. If you get MEM scores that are less than whatever your threshold is, and you can set the threshold a little if you want, but if you just always do 0, that's a pretty unbiased way to do it. So I would agree with that assessment. MEM is a non, or at least significantly less user biased way of merging clusters. All right, so there's the completion of our HyperFinder. It started with CD8 versus interferon gamma. So no big surprise there. We saw that what stood out on the MEM scores. Why was this cluster so different from the rest? Interferon gamma +8 or whatever it was, +5 or something like that. CD8 +5. So the two most important things that MEM identified as being different, independently HyperFinder said, well, if we start there, that's a great way to find that cluster. And then we can revise it. Let's get rid of the dead cells. These were CD3 positive. Let's essentially make a singlets gate. And then let's finish it off by, it's going to be most of the CD38 versus interferon gamma cells, but not all. Does a pretty good job of then isolating that last little bit of the population. Gets us to an F measure score of 0.94. It says done. We can just close this. And there's our HyperFinder gates. So if I was working with FACS Course, I save this workspace, I open it in FACS Course, and it imports the gates. If I'm working in Diva, I export to BD FACS Diva. You'll need to merge this with an existing Diva experiment file. Fine, so that's what the selection is at the top. Down below here is what am I interested in exporting. Maybe I would turn off everything at the single cell manual gate level below. Maybe I would turn off these cluster explorer populations and just pick my hi-fi gates and hit go and export that to FACS Diva. So that is how you can use HyperFinder to do computational sorting, and it's now built in, which saves us the trouble of having to work with a plugin there. Okay, that is about the end of our time. I've hit quad merging, I think quick, I've hit UMAPs. Let's remove a bubble. So if anyone has the ability to stay a couple minutes longer, just double clicking on a file here, if I had some amount of time I wanted to remove, I could just put time on an axis, look at the plot, and if you see some janky spots at the end where you see a lot of white space, you see a lot of issues, we could just draw a big rectangular gate and be like, okay, I want everything but the end. Cool. So that's one way to do it. The better way is we could use one of our quality control algorithms. I could pop on this population, go to the plugins. There are a few different options for quality control. My personal favorite is FlowAI. The reason for that is it does 3 different checks of quality. It looks for signal stability, it looks for dynamic range, it looks for flow rate. If any of those things vary, it just gates out the small interval of time that is variable. It also gives you a nice report. So let's run it here. Which parameters are of interest is pretty much always question 1. And in the case of FlowAI, it's going to be, you want to pick all of them, and you want to pick all the uncompensated ones. Theoretically should be the same for comped versus uncomped, but uncomped means that you are not going to have some compensation or cause some kind of thing that we interpret as a flow rate issue. So can't hurt to just pick the uncomped. Do you want to do all those checks, 2 of those checks, 1 of those checks? Take your pick. Generally, all checks is the way to go. You've got some tunables down here. I usually go with the defaults other than the change point penalty is how big does a spike in the signal have to be called a change point and a moment where we should worry and should we gate things out. FlowAI, I think, is a little bit on the strict side by default. I make that bigger. I usually go 500, and I also allow it to remove outliers, which just means that if we see a spike in the signal, typically what FlowAI does is say, well, that is a change point, and I should get rid of either all the cells before or after, whichever one has less, because they're not comparable. If you check the remove outlier box, it can look to see, does the signal come back to normal? Was that just an outlier, like 1/10 of a second? Always check that one on. It'll save you a bunch of data. I say, okay. It's going to think for like 30 seconds. I have this workspace saved in this 1010 demo folder here. And it is called, I think, 1010 demo, which has now a folder somewhere in it. Oh, there we go. There we go. FlowAI. Okay, so we're running FlowAI on this file. It automatically creates that FlowAI file. Oops, something went wrong. Huh, awesome. Demo syndrome. Anyway, and did it work at some point? No, it just failed. Probably some combination of stuff I just modified, did something funky. So I'm just going to try to redo that on, I think I have the same file, or at least a file from the same experiment just sitting here. Workspace, plugins, FlowAI, just sitting here, not futzed with. And so we can get some results out. And that is going to then, where did I save this one to? Is it on the desktop? It should just be called copy transform. Copy transform. Okay, so that now just automatically made a FlowAI folder under the folder with the same name as the workspace. All the stuff generally automatically creates. You get a subfolder with the name of each one of the samples that it runs on. And here is your report. I'm going to double click on that. Okay, so what FlowAI has done, and you can see over here in FlowJo, it came back with a good events and a bad events gate. They are exactly what they sound like they are. And it produced those by first looking at the flow rate, saying, okay, there's this spike at the beginning, startup. Put a green circle on that. Say that 1/10 of a second's unreliable. Sure. Rest of it's pretty good, drops down a little, rises up a little. So it takes out a few moments here and there where the flow rate looks a little sketchy. Here is the signal. Everywhere it's yellow, it kept. So in this data file, it kept basically everything. If you would see like a big spike, then you might see like a circle on one of these just 1/10 of a second periods that says, okay, take that bit out. It looked unstable for one of the signals. And then it goes down and checks the dynamic range. So essentially events pinned to the topmost or lowermost bin, because things that are in the top bin, just everything that was beyond the range of detection by cytometer all gets lumped in there. We don't trust it. Same thing basically for low and everything below the range of detection. And it says this many events fell into each of those bins, and we just remove them. So if you're worried about bubbles or clogs or sketchy flow rate or a detector that went bad or dirty or something like that, this tool is trying to get all of that. And it gives you that report that you could look at. It gives you the gates that you can work on. After you run it, you can just delete the FlowAI icon. And if we wanted to check the data out, we can just even overlay good events versus bad events, pop this versus time. maybe not a count, but just pick a parameter and say, okay, so we can see, let me actually change the colors to make that a little easier. Let's do just gray for background, red for bad. We'll just flip them. Here's those 3 or so, and then the beginning moments where the flow rate looked off, and then a smattering of red dots here and there are events that were probably stuck to one of the dynamic ranges. So I think that's a good way to look for any kind of technical variant that happened during acquisition. So there we go, checking for bubbles. Okay, we are 5 minutes after. The chat looks good. The QA looks good. So I think, thank you for joining. We've hit that point of the webinar. Thank you for joining. Really happy that you came to see what's new in 1010. Give it a try. I hope you use it. Happy to hear feedback all the time. john.flowjo@bd.com if you want to let me know if things work, don't work, or good for you. Anyway, happy flowing, everybody. Oh, I saw a question. What do you do if you receive an X when opening FlowJo files, like an X over here? It means FlowJo doesn't know where that file is. You should right-click on that file and find wherever it is. Search for FCS files. There it is. Search for FCS files. Point to where it has been moved to, and that should reconnect it. All right, all. Have a good one. Thank you, Kurt. Appreciate it.